Hello, everyone. Welcome to this presentation from the National Center for High Performance Computing, Taiwan. Today, we will explore how we are combining big data, HPC, and AI agents to drive the next wave of biomedical innovation. We introduce Agentic HPC for autonomous 16S rRNA microbiome analysis. By leveraging cloud and air-gapped HPC agents, we can securely execute entire QIIME-2 workflows, from raw sequence data to validated scientific reports with local LLM inference protecting data privacy. Currently, bioinformatics pipelines still rely heavily on fragile, manual command line execution. Agentic HPC changes this paradigm. Instead of manually writing scripts and troubleshooting errors, researchers define high-level scientific goals, and our autonomous agent manages the execution and self-correction. The 16S rRNA pipeline is an ideal stress test. It requires 9 complex steps from raw FASTQ import to taxonomic assignment. Each step involves critical decisions, like quality trimming and sampling depth, requiring both computational execution and deep biological understanding. Real-world science is full of friction, such as outdated SOPs or software version mismatches. To be truly useful, an agent must operate in a closed loop, planning, executing, diagnosing errors, and updating parameters, all while respecting strict institutional security boundaries. Our proof of concept evaluated 6 critical dimensions: autonomous execution, consistency across different HPC architectures, real-time error correction, private local inference, biological validity, and performance inside a completely isolated, air-gapped sandbox environment. We tested 3 agent architectures executing the same microbiome workflow. The first used a cloud-connected API, while the other 2 utilized on-premises LLMs, specifically Minimax and NVIDIA Nematron, deployed entirely within NCHC's secure GPU infrastructure to keep data private. In our air-gapped sandbox, we protect sensitive biomedical data across 5 layers, from raw inputs to the final report. With telemetry disabled and network traffic blocked, all model inferences and computations remain entirely inside the secure NCHC HPC boundary. Here is how the agents operate. The researcher simply defines the goal. The agent then plans the workflow, submits Slurm jobs, parses logs, and successfully troubleshoots any errors. All 3 of our tested agents autonomously completed this end-to-end process. Remarkably, all 3 agent architectures reproduced identical biological patterns. The resulting beta diversity plots clearly separated the samples by body site, showing high similarity between left and right palms, validating the scientific accuracy of our autonomous runs. Agentic HPC fundamentally shifts the user experience from routine command execution to scientific intent. It delivers scientific validity, operational efficiency through full provenance tracking, ironclad data privacy, and keeps the human expert in the loop to review and validate results. While this POC proves feasibility, our next steps include establishing a controlled benchmark with larger datasets. Over the next year, we will introduce stronger safety guardrails, including human approval checkpoints, strict sandbox environments, and multi-omics workflow expansion. Ultimately, Agentic HPC transforms secure computing into an autonomous scientific workbench. By combining local LLMs, Slurm scheduling, and robust bioinformatics tools, we allow researchers to focus on analyzing results and designing their next breakthrough experiments. On behalf of the National Center for High Performance Computing in Taiwan, we appreciate your interest in our research. We look forward to collaborating with the global community to build a more secure, AI-driven future for biomedical science.